Dr. Charles Serhan, and Charles is the Gelman professor at Harvard Medical School and also the director of the Center for Experimental Therapeutics at Brigham Women's Hospital. And Charles is going to be talking to us about resolvins and polyunsaturated fatty acids in the setting of infectious inflammatory disease. So Charles, could I hand over to you, please, and share the screen?
Hello, everyone. I'd like to begin by thanking the organizers and Professor Phil Kaldor for the opportunity to present our research to you today in this very exciting session. My institution requires that I show this slide. I have no conflict of interest for this presentation. I do serve on several scientific advisory boards listed here. My laboratory focuses on what controls excessive inflammation and infections. And of course, we love lipids. And the outline for today's presentation is to give you a little background on active resolution of inflammation and the structural elucidation of the resolvins, protectins, and maresins, which have come to be known today as the specialized proresolving mediators. And then a little bit of our functional decoding metabolomics research on deep vein thrombosis, and then some work from around the world of the laboratories and their contributions on human COVID and SPMs. So if we look at the acute inflammatory response from the textbook perspective, it looks like a decision path for acute inflammation, which is protective, to either become chronic, go to wound healing, or to resolve. The cardinal signs of inflammation are evoked by the classic chemical mediators, the prostaglandins and leukotrienes, all geared in this acute inflammatory response to protect us from invading organisms. What we found is that during self-limited, namely responses that resolve on their own, that rather than being a passive response to resolution, it's actually a biochemically active and cellular active process orchestrated in part by resolvins and a whole superfamily of lipid mediators that are produced during this phase. This slide illustrates self-limited or self-resolving inflammatory exudates. And of course, the ideal outcome for an acute inflammatory response is complete resolution. We've kept our eye on what's called PMN swarming, which we described earlier, where neutrophils move along a chemotactic gradient of leukotriene B4, like sharks smelling red blood cells, to swarm and then devour microbes to protect us from invasion. And we've used models of acute inflammation that are textbook and that there's rapid, within seconds to minutes, edema formation, followed by neutrophilic infiltration where they reach a max and then they are lost from the site. You can think about this as a pustule. And then the non-phlogistic, non-fever recruiting of monocytes and macrophages to the site. And we've used, for the systems approach, collection of pus, if you will, to interrogate on the back of mice initially. And it's within this time frame that we saw what we called lipid-mediated class switching, where the SPMs are turned on, first the lipoxins, and then the resolvins, protectins, and maresins. So the beginning signals the end. These are the two main functional responses we used for resolution that were used for the structural elucidation of the mediators isolated from pus. And that is the cessation of neutrophilic infiltration, the stopping of neutrophils coming to the site. They need to stop. And then when cells become apoptotic after defending us, the PMN, they are phagocytized by macrophages in a process called efferocytosis, and this is enhanced by SPMs, as well as upregulation of phagocytosis, killing of microbes and bacteria. To pinpoint these biological responses in vivo, we used resolution indices that we introduced. I should point out that traditional non-steroidal anti-inflammatory drugs do not stimulate resolution, and it took us some time to recognize that pro-resolution mechanisms were not equivalent to anti-inflammatory mechanisms. The specialized pro-resolving mediators are produced from n-3 essential fatty acids, primarily EPA for the E-series resolvins, DHA for the D-series resolvins, and the protectins formed from 17-hydroperoxide DHA intermediate, a lipoxygenase pathway product, and then the maresins, the macrophage mediators of resolution from DHA and macrophages. We did the complete stereochemistry of each of the mediators, which you'll see is important, as well as their total organic synthesis to confirm biosynthesis and function. We were also able, with the resolvins in hand, to introduce criteria for pro-resolving actions, and the most important of which in vivo is the shortening of resolution interval, R sub I, and of course, reduction in pain. This slide illustrates the SPMs and the principal cell types that we know in human systems to produce SPMs. Apoptotic PMN, as a lion's share, as well as M2 macrophages and a number of other cell types when they interact with neighboring cells. The main tissues we see and others have documented resolvins in lung, spleen, marrow, and lymph nodes. This slide illustrates the potent actions of the specialized pro-resolving mediators in stimulating as agonists the resolution of inflammation by the signs of resolution. And they are in the potency range of leukotrienes, and they're more potent than prostaglandins, the classic pro-inflammatory mediators or cardinal signs of inflammation. They have considerable translational potential, and they've been confirmed, their actions in many animal models. They're 100 to 1,000 times more potent in animal models than non-steroidal anti-inflammatory drugs. In inflammation models, tissue protection, infection, neuroinflammation and pain, wound healing, and even cancer models of stimulating the resolution of inflammation around cancers, reduce cancer burden. With the complete stereochemistry of each of the pro-resolving mediators in hand, we were able to identify some of the first GPCR cell surface receptors that are pro-resolving receptors, illustrated here. The lipoxin A4, the resolvin D1 receptor. They're each stereoselective for activating pro-resolving signals, and very recently the MAR1 or the maresin 1 receptor, LGR6, which has already been confirmed by other groups. So how do SPMs work? They have a novel mechanism of action, as illustrated here. They shorten the resolution time. For example, resolvin D1 shortens that resolution time from 20 to 12 hours in pico to nanogram range. They downregulate the cytokines and chemokines, as well as the eicosanoids at both the transcriptional level and the biosynthesis level. So in all, the SPMs have been shown to limit the magnitude and the duration of acute inflammatory responses, and they are not immunosuppressive. To give you an idea of some of the potency, this is in a sepsis model, polymicrobial sepsis model, and you see that Resolvin D2 at as little as 10 picograms per mouse reduces neutrophilic infiltration. And on the right of this slide, Resolvin D1 and D5 increases in phagocytosis and killing of E. coli in the 0.11 nanogram, 1 nanomolar range. We have introduced auto extractor in our lab and are using a targeted lipid mediator metabolomics approach, illustrated here, that relies on LC-MS-MS. We have introduced spectral libraries for all of the proresolving mediators, which are also commercially available now. And we quantitate not everything, but have focused on these pathways in a targeted bioactive metabolomes, and then principal component analysis, which we think will be useful in personalized and precision therapeutics and nutrition in the future. Using this system, and just before COVID, we looked into deep vein thrombosis. So what is the temporal relationship of lipid mediators in this deep vein thrombosis, known to be a problem in pulmonary embolism? And as we learned during COVID, coagulopathies proved to be an important component of the COVID-19 infection. And so with Denisa Wagner's group, an expert in thrombosis and intravital microscopy, along with Dia Jepanovic from her group and Charlotte Jovine, we examined this occlusion of stenosis model, which is nicely illustrated here, out to 21 days, where they get a complete occlusion, a blockage of the vessel, if you will, and it begins to resolve by day 21. And in this, we systematically analyzed by LC-MS profiling, just showing you the lipid mediators produced at each time point in vivo. We found that there's early phase, as one may expect, the activation of prostaglandins and thromboxanes that moves to a middle phase of lipid mediators where the resolvins begin to get turned on and then remain elevated out to 14 days in this animal model. Given that as a backdrop, we decided to treat the deep vein thrombosis with a constellation of pro-resolving mediators that we found that are being produced in this model. And this is a Doppler ultrasound imaging, and you can see time-dependent changes in the diameter in that DVT model that is dramatically reduced with SPMs on board. So the thrombus size is sharply decreased when Resolvin D1, D4, MAR1, and LXA4 are all given together in this scheme illustrated in that timeline. So we worked out the biosynthesis of Resolvin D4 earlier, its total organic synthesis, and the matching of these physical properties and of the synthetic material along with the biologic, as we have for each of the pro-resolving mediators, and added back solely Resolvin D4 in the system and found that it reduced the thrombus burden by stimulating resolution of thrombus, increasing apoptotic cells, and therefore Resolvin D4 attenuated the severity of the pathologic thrombus. So if we have this as a biological set of actions of these mediators produced from n-3 essential fatty acids, which are not immunosuppressive, is there a possible link to COVID-19 treatment? Well, As I emphasized, this eicosanoid and cytokine storm is controlled by the Resolvins. We introduced with our colleagues in this review the notion, and now many others, that SPMs can be used to control the airway inflammation and the systemic impact of the cytokine storms. Well, we're gratified to see this publication recently from Antonio Ritucci and his group, Mario Romano, with human cystic fibrosis macrophages exposed to the spike protein, which turns on chemokine and cytokine pro-inflammatory response. That was sharply reduced with Resolvin D1 and D2. And you can see the evidence of their gel, and this was recently reported in this publication. Very exciting. This publication looks using the LC-MS-MS method, similar to the one that I outlined to you. Ben Schwartz looked at the peripheral blood of COVID patients that were severe or moderate and found that prostaglandins and leukotrienes are produced in high levels in these severe patients, whereas resolvins are produced in the moderate COVID-19. In their dot plot from their paper, their heat maps show that resolvin D4 increases in the moderate COVID-19 and resolvin E3 from EPA in moderate drops during severity. So there are dynamic changes in the lipid mediators and most importantly, an increase in the proresolving mediators in moderate and a dramatic increase in the leukotrienes in the severe. This is an exciting report from Nicolas Froment's group in Laval in Canada, where they looked at bronchial lavages from around the world, multi-center sites, and found that in the human bronchial alveolar lavage, they could identify using LC-MS-MS, the pro-resolving mediators, concomitantly with the pro-inflammatory mediators in the bronchial lavages. So demonstrating that they are produced in human lung and maybe could be enhanced to counterbalance the excessive inflammation. There have been a number of studies showing that SPM increase in human peripheral blood on supplementation from other groups. This shows a 10-week study with 3 grams per day in this double-blind randomized crossover. Also, time and dose-dependent increases have been seen by this and other groups, just at the beginning of this line of research. So in summation, we've shown you that SPMs carry a new biological action, namely pro-resolving function, that are agonists to stimulate the cardinal signs of resolution. They taught us that resolution of inflammation is an active process, and this was a structural elucidation of dark matter in the universe, if you will. We're using functional decoding metabolomics, SPM RVD4, for example, identified here in a DVT model that reduces coagulopathies by stimulating resolution of thrombus in vivo, at least in animal models. And I've shown you results suggesting a role for SPMs in human COVID-19 disease and recent results from supplementation indicating that SPMs can be increased in humans. So with their receptors, we think that this could help in the control of excessive inflammation and infection via resolution physiology and resolution pharmacology. And it's most important for me to thank my colleagues, shown here in the upper left in my laboratory, the members of my NIH-supported program project team here, long-standing collaborations with Dr. Nikos Putasis, supported by NIH and on the stereochemistry, and Bruce Levy and Matthew Spite, Nan Chang, and others on our efforts to understand the resolution of inflammation. So with that, I thank you.
Thank you for that very elegant presentation, Charles. Again, we have time for questions, which you can post on the question board. While we're waiting for people to do that, maybe I could just kickstart some questions, Charles, by asking a little bit about the coagulopathy elements of the resolvins. To what extent does this effect involve interactions with actin and actin polymerization? Is this something that's important? Because what we tend to see in acute diseases or any sort of inflammatory responses, that some of the proteins that would be naturally act as inhibitors of actin coagulation are lost. So how do the resolvins compensate for that?
Thank you, Martin. Yes, ultimately, it's the role of the macrophage here is resolving these intravascular clots by phagocytizing them and clearing them. So they're actually stimulating the resolution of clot and in a deep vein thrombosis model. And when added back, the very exciting finding is that when we added back resolvin D4 in this mouse model, that turned on the production of other resolvins, ones that are known to prevent platelet aggregation and counterregulate other procoagulant eicosanoids. So there are multiple sites in which the proresolving mediators can help to regulate or stimulate the resolution of intravascular clot, including acting on vascular endothelial cells to produce substances like NO, etc.
Interesting. Further questions? I guess the other question I had, in the absence of any questions appear on the bulletin board at the moment, is again, it's the same question I had for Phil, really, which is to what, with COVID-19, we're seeing this as a very age-centric disease in a way. Is there evidence for aging effects with resolvins where you're seeing less effective function with age, or is this something that are there potential dysregulations occurring in older populations?
Right. Yes. So the studies that I'm aware of, one's from my laboratory, and then several others have looked at the mouse models of young versus aged, the NIH mouse model. And in that setting, there's excessive acute inflammatory response. And what we found is there's an actual absence of the resolution mediators. They're diminished in aged mice, but we could replenish them, which we thought was rather exciting, and turn back, if you will, the age-dependent impact on resolution and counter-regulate the excessive pro-inflammatory response seen in those mice. Now, that has not been translated yet to human studies, but there are very strong results on differences between men and women published by Oliver Wertz's group in the production of leukotrienes, and there are clear differences in women and men that I would venture to say would persist into age. Yeah, and this will have effectiveness on those drugs that are known to regulate leukotriene production.
Yeah, it's an interesting point, given that inflammation as a whole tends to sort of be enhanced with aging. So you have this increased dependency on anti-inflammatory steroids, which are not necessarily particularly good for older people as well. So this is an interesting new angle on that.
I agree entirely, and it brings up the point that we really need to have precision nutrition here to enable the innate immune system and its resolution, but also the opportunity for not a one-size-fits-all as we use today for non-steroidals and other anti-inflammatory agents, but very, I would say, personalized.
Yeah, that's a good point. We have time just for one quick question, because a question has been posted. This is from Gretchen Vanis. She says, you mentioned human supplementation increases SPMs. Is that the supplementation with EPA and DHEA from fish oil or from other supplementation?
Thank you. Yes, as I pointed out several recent studies that have shown with EPA and DHA increased peripheral blood production of a panel of SPMs. I should point out that not all SPMs appear in peripheral blood. And so we need to still think about these as local mediators, and what we're seeing in the peripheral blood may just be on their way out before excretion.
Okay, thank you, Charles. I think in the interest of time, we will move on to the next speaker, but thank you again for your presentation.